today I'm going to be focusing on this enabling fast and accurate real-time analysis, analyzing the raw nanopore signal data. And I'm going to mainly be presenting RawHash and RawHash2 papers. So RawHash was presented at ISMB last year. So you can use these QR codes to access the paper and the source code. So let me give you a little bit background then before I start describing RawHash. So when we look at the raw data that these sequencing machines generating, so they all generate different kind of data, right? But they all generate raw sequencing data at one point before they are converted to the base code sequences. And today we're going to be mainly looking at this nanopore sequencing technology, which generates raw electrical signals. And I'll try to give the reasons for that. But mainly the nanopore sequencing is a widely used sequencing technology in our field. So it can produce long reads up to 2 million nucleotides. It provides opportunities for portable sequencing because there are these devices that can literally fit in your palm, so you can carry it with you and then do your portable analysis. It is relatively cost effective. And there are also some more unique features such as real-time analysis that I'm going to be covering now. So if you're not familiar with how nanopore sequencing works, so you can imagine a 3-layer solution here. I'm not sure which part is maybe it's easier. So you can imagine a 3-layer solution here. So on the top layer, we have a negatively charged region. And on the bottom layer, we have a positively charged region. And in the middle, we have a particle membrane where this pore that we call nanopore is sitting. So what's happening here is that since the DNA is also negatively charged, so this DNA is literally moving from the negatively charged side to the positively charged side. And as it moves through, it creates ionic current disruptions over there. And these disruptions are measured and reported as electrical signals. And these disruptions are significant enough for us to tell each base, each nucleotide in this DNA molecule, so that these electrical signals are then analyzed. Let's say we could be translating these electrical signals to ATGCs again, using a computational approach called base calling, or we could also directly analyze these raw electrical signals without base calling them. But the opportunity here is that these electrical signals are reported in real time. So this means that we can also start analyzing these electrical signals in real time while they are being generated. And one very important feature of nanopore sequencing is, based on your analysis, you can also give feedback to the sequencer and telling, for example, if the read that you're currently sequencing is somewhat useless to you, or basically you've done enough analysis for that particular read, you can tell the sequencer to stop sequencing that read at runtime. So this is basically very advantageous, and I'm going to be describing those benefits now. So there are 2 main benefits with real-time analysis. One benefit is, in your most common case, what you would do, you would first sequence, get your data, and then do your analysis, right? And then this is the entire time you would spend. But with real-time analysis, you can sort of overlap the sequencing time with the analysis time. And then this would basically reduce the overall latency of genome analysis. So this is one advantage. The second advantage is that, again, in the usual case, we would be completely sequencing reads. So you don't have the option to tell the sequencer, oh, I want to stop sequencing that read in the middle, let's say, so that you can save maybe from sequencing time and cost. But with real-time and nanopore sequencing analysis, we can do that. We can partially sequence the read. So this means that we can reduce the sequencing time a lot and also potentially the cost by doing so. But of course, there are challenges for real-time analysis. One challenge is, of course, we need to match the data generation speed. Our computational approach should match the data generation speed. Otherwise, it's not real-time. The second challenge is that we really want to make these rejection decisions or ejection decisions extremely quickly, so that we don't sequence unnecessarily, basically. Of course, also we also want to do accurate analysis, right? Our analysis needs to be accurate, that's obvious. And also for portable sequencing reasons, we want our analysis to be power efficient, right? That's another challenge that we need to keep in mind. And so I already told you that we can perform raw signal analysis without base calling. And this is, again, a quick background on this. So you can here assume that at a time within this nanopore, there are K-many nucleotides basically included within this pore. So this K-many nucleotides is usually from 6 to 9 mer, depending on the technology that you're using, depending on the chemistry that you're using. And so this is basically is referring to a K-mer, right? So, and we also have this term that we call event. So this is basically this electrical signal that is generated from nanopore sequencing. And this event corresponds to a particular segment in the raw signal. So what this event is basically the part of the raw signal that we generated while we're sequencing that particular K-mer over there. So the events are basically corresponding to the K-mers, the sequencing of K-mers. And then whenever basically a new base is coming in to this nanopore and another one is coming out, there's usually an abrupt single change in the signal that is reported. And usually statistical methods are used to identify these abrupt changes. And basically then based on these markings or the abrupt changes that we find using statistical methods, we calculate the average, let's say the signal value over there, which then is referred as the event value. That corresponds to basically the signal value that we would have when sequencing a particular K-mer. But there is one critical observation here, which is when we're sequencing identical K-mers, we don't generate exactly the same signal, but we generate similar signals, let's say. So that's an important observation that we're going to use later on. So with that, I'll then start describing RawHash. So the problem that we see is the real-time analysis of raw nanopore signals is either inefficient or inaccurate, especially for large genomes. And our goal with RawHash was to enable this fast and accurate analysis in real time, specifically also for large genomes. And we make two key contributions in RawHash. So RawHash provides the first hash-based search mechanism for mapping raw signals, for mapping raw nanopore signals. And the second is we introduce a method that we call the sequence central technique, which can dynamically stop the entire sequencing run, basically not a sequencing of a single read, but the entire run of sequencing if basically further sequencing is unnecessary for the type of analysis that you're using. And we look at relative abundance estimation for that analysis, and I'm going to be talking about this more in the coming slides. And based on basically across 3 use cases and different data sets from varying sizes, including a human genome, we find that our work essentially outperforms the existing state-of-the-art works by 27x, 19x, and 4x in terms of the throughput, computational throughput. And also RawHash provides the most accurate raw signal mapping for all data sets. And when we look at basically the benefits of the sequence central technique, we also see that we can reduce the sequencing time and cost by 15x, basically, when this technique is enabled. So there are 2 solutions for performing real-time analysis. One, as I said, we could be doing base calling, and then you could be analyzing your base call data as you would do in real time. So this is good because it is usually a more accurate approach, right? This is considered as the golden standard for the best accuracy. But this is also extremely costly and power hungry. So you are usually using basically these expensive deep neural networks with your GPU and so on. This is basically limiting your scalability. So the second approach is mapping signals to the reference genomes without base calling them. And this is basically is beneficial for 2 reasons. Raw signals contain richer information than just bases. You can be inferring the methylation information, for example, as well directly from raw signals. And then it also provides efficient analysis with better scalability and portability. So I'll skip this slide because I already covered this one. But basically, when we look at the problem in these approaches that perform the mapping of raw signals, it's actually about the scalability for larger genomes. So when you have your raw signal, and then when you're mapping to a small region, small reference genome, what you usually have is that you have fewer candidate regions, right, in small genomes. So this means that you can somehow do some accurate mapping, and also you can provide high throughput because the problem size is not really large. But when you basically start dealing with the larger genomes, for example, a human genome, then this means that you have substantial larger number of regions that you need to check per read, basically. So the cost of computation per read increases substantially with the larger genome. So this leads to two problems. One problem is there are probabilistic approaches called uncalled. And when you're working with many regions, specifically with repetitive regions, this particular work cannot perform accurate mapping because it cannot decide accurately from these too many regions. So there is another work that's called SIGMAP. It performs distance calculation to identify the similarities and to perform mapping. But again, when you're working with a larger, let's say, reference genome, that sort of distance calculation becomes too costly again, so that the analysis itself stops becoming real time. So then we find that the existing solutions are either inaccurate or inefficient for large genomes. And our goal is to essentially solve this problem by enabling fast and accurate analysis. So to this end, we propose RawHash. We make two key contributions. We provide the first hash-based search mechanism, and also we propose the sequence until technique. So I'll first describe the hash-based search mechanism that we provide in RawHash. So remember the observation that I already mentioned earlier, right? So when you have, let's say, when you're sequencing the identical nucleotides, assume that you're sequencing a particular k-mer twice, right? So what's going to happen is that you're going to generate, let's say, two different signals, and these signals are not going to be exactly matching. But the observation is that these signals are going to be similar to each other. And the earlier work exploits this observation by calculating the distance between signals. And when you calculate distance, you can tell the similarity and so on. But we don't want to do this because it is costly. Rather, what we want to do, we want to somehow generate hash values from these signals. And these hash values then can be used for fast match purposes, right? But then the question is, there are basically two challenges. And one question is, how can we generate the same hash value from different signals or similar enough signals, right? So we need to be able to do that. The second challenge is that we also want to accurately find as few similar regions as possible, right? So that we're not really looking at too many regions at a time, but just only a few regions, even if it is a large genome. So this is the overview of RawHash. So we start with converting the reference genome into signal space as well in our indexing. And similarly, again, we convert the raw electrical signals to the event values that I described you earlier. So we complete the work on the signal space, including the reference genome. And then to eliminate some noises, we perform quantization, binning, and so on. And then we generate hash values from these noise-free, almost noise-free values, so that we can store these hash values in the hash table for reference genome, and then later when we're generating the hash values from the reads, we can create the hash table so that we can do our usual chaining and mapping steps that we do in Minimap2, for example, right? And the steps here on the left is an offline step, is just a one-time task for each reference genome, and the right step, the right step would be here the real-time analysis part. So I'll start with the reference to and also signal to event conversion. As I told you, we're converting the reference genomes to signals, right? And the way that we're doing it is essentially we essentially take the k-mers from the reference genome, all overlapping k-mers from the reference genome, but the size of the k is really important here because it is really dependent on the nanopore sequencing, nanopore chemistry that you're using. So remember, I told you, like, usually it contains the 6 mers or 9 mers within a nanopore. So you should be basically taking the correct k-mer size here when you're converting your reference genome to signal. And then what we do is that there are these lookup tables that tells us, given a k-mer, what is basically, so these k-mer models are pre-constructed based on the nanopore, basically the characteristics of the nanopore. But what happens is that given a k-mer, it tells us what is its expected signal over here. And then we can do this for all k-mers and then do normalization and so on, such that rather than basically working with k-mers, we work with the signal values of these k-mers. So this is basically pretty simple. This is a one-to-one conversion of the k-mers to the signals. Then we also convert, let's say, the raw signals to events, because we just converted the reference genome again to events from their k-mers. And to do this, what we simply do is that given a signal, we perform this segmentation, which is a statistical test that identifies these abrupt changes in the signal, which is our events. And then we calculated mean and normalization and so on. But the values that we're getting at the end is somewhat comparable to the values that we generated from the reference genome. So now that we have these basically consecutive events, which are supposed to be consecutive k-mers, so the question is, can we directly match these signals, or can we directly match these events to each other to identify similarities? So for that, I'll cover the quantization step in our work. Again, I'll remind you the observation, which is basically that there are slight differences in raw signals, right, from identical k-mers. So this means that when you're sequencing this k-mer, you may end up generating different values, but they are going to be similar to each other. So you cannot simply try to match these values to each other, because you're not going to be able to match them, right, with a quick search. Rather, what we do is that we simply quantize or bucket or bin these values. There are different terms for that, to essentially reduce the value space, maybe from like 1,000 different values to around 32 different values, because here we use 5 bits over there. But the idea is basically binning over there, right? So that basically these values that are closer to each other can be binned into the same values, so that we can reduce the noise effect from these signals. So this is quantization. And then the last step would be generating the hash value. But there is one more challenge that we need to solve, which is basically about the length of these events, right? So when you try to match a 6-mer in the reference genome, you're going to find maybe thousands or millions of matches, right, in the reference genome. But we just quantize that k-mer, so we cannot simply use this 5-bit value or this particular small value just for matching purposes. So we need to somehow increase that k-mer size, basically. And the way that we do it is we assume that the consecutive events are consecutive k-mers. So we can somehow preserve the order information by essentially packing all these different quantized values together. Maybe we may have like 5 or 6 quantized values here. And then when you pack them together, what you end up getting is basically a huge and distinct enough hash value, which can be used for matching purposes, because this is already long enough and unique enough, hopefully. So this is basically how we're generating the hash value from raw signals. And then to basically wrap up all the steps that we do for real-time analysis, here we construct the hash value from the reference genome by converting it to signal. And then what we do is that we store these hash values and their positions in the hash table, right? This is indexing. And then again, in real time, so this is stored. And then in real time, what happens is that we similarly generate the hash value again from raw signals while the raw signals are being generated. And then we query the hash table. And if there are some matching positions, we perform chaining and mapping and so on. And based on the mapping information that we generate, we ask the question continuously whether we should keep mapping, meaning whether we should keep sequencing this read or not. And if the answer is yes, we simply retrieve the next chunk of data, do all of these steps again, and then do this checking again, and then so on. But if the answer is somehow no, then we tell the sequencer to stop sequencing that particular read using the features that are known as read until or run until. Basically, this is the entire real-time analysis using RawHash. So let me see how I'm doing with time. OK, I guess I have a little bit more time to cover the sequence until mechanism. So the sequence until mechanism is somewhat different than RawHash, but there's a good synergy, let's say, between two, because they can be used together for reducing the sequencing time and cost. So the problem that we're dealing here is that unnecessary sequencing obviously wastes time, power, and money, and we don't want that, right? And the key idea in sequence until is to dynamically decide if further sequencing of the entire sample is necessary to achieve high accuracy, right? That's the key question and the idea that we're trying to answer. And then if it is not necessary, we can stop the sequencing early without sacrificing the accuracy. And the potential benefits, of course, the significant reduction in sequencing time and cost. And then for the analysis purposes, we look at a particular use case, which is a relative abundance estimation. And then we try to perform relative abundance estimation without sequencing the entire sample. And there are basically a few key steps to achieve this. So what we do is that we continuously generate relative abundance estimation after sequencing of every n reads. For example, we just sequenced, 50 reads, and then based on that 50 reads, we do some relative abundance estimation, right? Now this is your estimation. And then we basically keep doing this, and then we always keep the last t estimations that we had previously, right? So this is the last t estimations as we keep sequencing. And then what we do is that as soon as we have t estimations, or as soon as we have a window of t estimations, we ask the question whether there is an outlier in our estimations. So if there's an absence of an outlier, then this means that we are performing relative abundance estimation pretty consistently. So this means that the further sequencing is likely also to generate the consistent results. So then this means that we don't really have to keep sequencing the entire sample. Rather, we can stop the entire sequencing run, because when we looked at the past window of t estimations, our estimations basically doesn't change much. So we have a pretty good confidence that it won't change with the further sequencing as well. And basically, if there's an outlier, then this means that the sequencing keeps changing the results substantially. So this means that we just need to keep sequencing it, and then we do it basically until we reach to this decision point. So this is sequence until, basically, and then we can somehow merge these two with RawHash and RawHash2. With just a single or two slides, I'll cover the optimizations that we make in RawHash2. So RawHash2 is a follow-up work on RawHash to improve its accuracy and performance. And what we bring up in RawHash2 is basically we have this new quantization mechanism that is more accurate and also more efficient. We also improve the chaining algorithm with more sensitive penalty scores. Our decision making for stopping the mapping process is now like a ML decision making. So we have this almost single layer, single neuron architecture over there that makes a more robust decisions. And also we integrate filters and minimizer sketching as well to achieve more space efficient, and also to reduce the seed matches for our analysis. And then if you want to basically imagine how the minimizer sketching could be integrated here, is basically when you have your hash value over here, what you could simply do is that now that we are in the hash value space, you could literally use all the existing sketching techniques out there, right? Strobmers, minimizers, or like any other sketching technique, and then sample your hash value based on that, and then generate your index accordingly, and so on. And then in the RawHash2, we integrate minimizers for that purpose. So I'll then quickly cover the evaluation. For RawHash2, we have two settings. We have RawHash2, the default setting. We have all the k-mers, and we also have RawHash2 minimizer with minimizer sketching. We compare to the state-of-the-art works, Uncode, SIGMAP, and our early work, RawHash. We have three use cases: read mapping, relative abundance estimation, and contamination analysis. And we have three evaluation metrics. One is throughput, which measures the basis process per second per CPU thread. We also look at the potential reduction in sequencing time and cost of each tool, and also we look at accuracy. And for accuracy, our baseline is the mapping of base code reads using Minimap2, and we look at the precision recall and F1 scores for accuracy. And for relative abundance estimation, we calculate the distance to the ground truth again, which is generated using Minimap2. We have 5 different data sets for read mapping, starting from a very small viral genome up until the human genome. For relative abundance estimation, we create a mock community by combining all of these data sets. And for contamination analysis, our sample includes the viral genome and also the human genome. And the question is basically whether the sample is contaminated by a viral genome or not. So we will first look at the throughput results, but I'll again remind you, the real-time analysis requires faster throughput, faster computation than the data generation speed, right? And the throughput that we get from a single pore is usually around 450 base pairs per second, right? This is the data generation speed per nanopore. And here we show the results for each tool. On the y-axis, we have the throughput numbers over here, and on the x-axis, we have the data sets and the use cases. And based on these results, we make two key observations. One observation is RawHash outperforms, improves the performance of throughput basically substantially compared to all these state-of-the-art works, including RawHash by 4x, its predecessor. And also, when we look at only the performance of RawHash 2 minimizer, it further improves the throughput results by 2.5 x compared to RawHash 2, because minimizer is a sampling mechanism. You have, let's say, a smaller problem to work on. So we also look at the average sequencing length, average sequence length when you're using each of these tools. And basically, here, we want to achieve fewer bases. We want to sequence fewer bases, because then this means that less unnecessary sequencing. And here, on this plot, the lower is better. And on the y-axis, we have the average, the sequence length that each tool requires before stopping the mapping or stopping the sequencing. And again, on the x-axis, we have the data sets. So we see that the RawHash 2 reduces the sequencing time and cost on average by 1.9x compared to Uncold and RawHash. And also, RawHash leads to the sequencing the least amount of bases for larger genomes. And what I mean by larger genomes is these ones except the viral and the E. coli. And for E. coli and viral, Uncold seems to be doing better than RawHash 2. And last, basically, when we look at the accuracy of read mapping, so what we're doing, we're mapping the reads to their coordinates in the chromosome, and we're literally looking at whether these mapping positions are correct or not compared to the min-map2 positions, right? And now for that, we have this, we measure the F1 score for read mapping, we measure the precision for contamination, and then we measure the distance for relative abundance estimation. And the observation that we make from these accuracy results is RawHash provides the most accurate read mapping for all data sets and for all use cases. And so I told you that RawHash 2 is fast. The question is it accurate? So when we look at the results, RawHash 2 minimizer provides an on-par accuracy with RawHash 2, but with, of course, substantial throughput benefits. So if you want to trade off between performance and accuracy, you have already your two answers over there. So I guess one last metric that I want to show is about the benefits of sequence sample, right? So in my opinion, this can be applied to all other tools other than RawHash, and we also have simulated results for this. And what we do with this evaluation is essentially we let the RawHash run by using the entire sample without the sequence sample capabilities, right? And then we make a relative abundance estimation using the entire sample. And then we activate the sequence sample mechanism, and then let the mechanism decide when to stop sequencing the entire run, depending on the outlier detection that I just told you. And then when we do this, we find that sequence sample stops after sequencing the 7% of the entire sample, right? So you don't know this number beforehand. You only know it after you do your analysis. And then after sequencing 7% of the sample, when we look at the distance of our estimation compared to the distance when using the entire sample, we look at the distance, we find that the distance is basically pretty low. So you can almost achieve the identical accuracy by only sequencing 7% of the entire sample. So this means that the sequence sample enables sequencing only using the 7% of the entire sample with high accuracy. And then we also simulate the benefits of sequence sample using Uncold, not just RawHash. We basically look at certain breakpoints, like what would be our relative abundance estimation at 1%, 10%, and so on. And then we find that actually we can reduce the sequencing time and cost by up to 100x using this sequence sample mechanism. So to conclude, RawHash provides the first hash-based search mechanism that is critical for fast and accurate analysis, especially for larger genomes. And the sequence sample can dynamically decide whether further sequencing is necessary for accurate analysis. We show, based on our analysis on different data sets and use cases, we find that RawHash is much faster than the earlier works. It's the most accurate mapping tool, and the sequencing tool reduces the sequencing time and cost. And we essentially, to wrap up and to some, I guess, give motivational points here, now that we can work in the hash value space, so this means that we can bring all the existing works in the base code domain that uses these hash values to perform certain analysis, right? We can bring all these analysis now to the raw nanopore signal space. And we also know that the indexing is very cheap. So this means that there are many future use cases that can be performed with on-the-fly index construction, perhaps. And so all this means that I believe we should rethink the algorithms to perform downstream analysis fully using raw signals, right? I don't see currently a reason why we shouldn't do or why we cannot do all the downstream analysis fully using downstream analysis. Maybe in the future, many tools will be developed for this purpose. So this was RawHash, and I'll quickly conclude again using a few other slides. So one of my conclusion remarks is I believe future is bright for raw signal analysis. So this work is not public yet, but it's going to be public soon. And one reason why I think it is bright, because now using these efficient hash-based search mechanisms, we can also perform overlapping in real time. And I think you already know what this means. So we can hopefully construct assemblies also in real time. In fact, we basically try to do it with the on-the-fly overlap construction. First, we look at the quality, or basically how many overlaps that we're sharing with the base code overlaps, right, using all these 3 data sets. And especially for smaller genomes, we see that the raw signal overlapping shares a large portion of it with the base code overlaps. But still, there are some unique overlaps that are unique to raw signal overlapping. And also there are some overlaps that are unique to base code overlapping, right? Then the question is like, are these unique overlaps perhaps more accurate, or like since we're not finding these unique overlaps in Minimap2, are we reducing the quality of or contiguity of the de novo assembly? And then what we do is that after constructing the overlaps, we also construct the assembly from these raw signal overlaps. And then we currently look at the contiguity of assemblies, and then we see that the answer for contiguity for almost all data sets is yes. Signal-based overlapping is currently less contiguous, but it gives some, I guess, exciting results, because in terms of the average contig length and the maximum contig length, we are already pretty close to, almost close to basically the base code overlapping. And we're basically just starting, I would say. And when we look at the human genome, actually, the surprising thing is, this is a very, let's say, low sequencing depth of coverage. What we observe is that with a low coverage, we can actually construct more contiguous and longer contigs using raw signals than base code. And we don't know the answer to that yet. So we'll look into it. We'll try to understand, perhaps maybe there are richer information. There's richer information in raw signal that leads to more contiguous assembly when the sequencing depth of coverage is low. There are basically new applications that we can keep working on, right? Especially with the overlapping information. So you could imagine, you could use the overlapping information for improving the base calling accuracy, right? You could give your base caller this overlapping information, and it could utilize it, right? And as I told you, I already told you that we can also perform full downstream analysis, fully using raw nanopore signals. I think that there's many work that can be done in this direction. And then we could also cooperate the raw signal analysis with base calling analysis, right, together, if this is useful. And also there are many, many more, I guess, exciting times and applications, keeping the hardware design in mind. And speaking of the hardware design, I basically tell the things are also happening in industry. So I'm pretty sure maybe you're already aware of this, the Dragon platform that Illumina provides. This is a full FPGA board that performs your entire genome analysis from mapping to variant calling in a faster way, right? And these are already included in your Nova and NextSec Illumina devices, right? So here they are basically also motivating the hardware acceleration and also the use of FPGAs for improving the speed of genome analysis. And there are also undergrad startups that simply basically exploit the GPUs and then accelerate. A very important application here is GATK, And then they accelerated so much that the NVIDIA actually buy them out, let's say. With simple ideas, you can even maybe perhaps earn millions of dollars, I assume. And also the NVIDIA and also other vendors are essentially looking at these important applications. Genomics is one of them. And then looking at what sort of operations are needed for these important applications, right? And dynamic programming calculation is one of them. And now the dynamic programming operations are now also natively supported in some of the GPUs, right, to accelerate certain applications, including perhaps read alignment and chaining. And also certain genomics companies are also collaborating with the tech companies, such as, for example, here Bionono we know that Bionono is collaborating with NVIDIA to accelerate their analysis. And now people are also looking at how can we basically design hardware from scratch to accelerate certain applications. Here, this is a Cerebrus wafer scale engine. This is an AI engine. It includes 2.6 trillions of transistors to accelerate AI ML operations. And then there are now also in-memory devices. So these are DRAMs that you can buy off the shelf, but they also have the capability of computing as well as basically storing the data. This is UpMem, and you can buy their modules and then do your analysis within the memory. And we are also a part of basically the effort that we call BioPIM, where we try to basically understand certain genomics applications and then try to see the opportunities, which of these applications we can use for in-memory computation, right? So which algorithms can we bring them to the in-memory space? And there are basically many partners in this group. There's Bilkent, there's IBM, ETH also, also some groups from France and so on. So we also publish and make certain materials available. So in case you want to learn more about basically the use of hardware and software together for fast genome analysis. So if you want to learn more about this, you can take a look at all these materials, and these slides will be available. Is already available on my website. We also organize workshops in this direction. Last year at Reconf, we organized the BioArch workshop where we had talks in this direction. And we also have courses, again, in this direction. Many students take these courses and then learn how to use hardware for accelerating, to accelerate the genome analysis. So in conclusion, so today we covered various recent ideas to accelerate genome analysis and also to analyze the genomes in ways that were not possible before. And in my opinion, enabling cost-effective, portable, fast, and accurate genome analysis has many implications. So basically, for example, what are the new applications to enable these unique benefits of, for example, raw signal analysis, right? And maybe next question is, of course, can we do even better? Can we sequence other types of biological data, maybe even amino acids, and then perform, basically do proteomics also in real time using raw signal data? And basically, to conclude, I think many future opportunities exist, especially with the new sequencing technologies and also especially with the new applications and use cases using the emerging hardware. So with that, I'll conclude my talk.